Today we're going to look at mitosis, and some of the questions that you get when you start thinking about mitosis is, you know, how do the little elephants grow up to be big elephants? So how does this take place? How do we have these changes? Why do animals shed their skin, and what does that do for them, and how does it happen? Well, the answer is mitosis. And mitosis, the process of asexual reproduction that begins after a sperm fertilizes an egg. So after the joining of the sperm and the egg, you see this cell that now is getting ready to go through mitosis. And again, in class we talked about how it's an asexual reproduction. It is not by sexual means. So this reproduction is going to happen without the sperm and egg because it's already happened. The sperm and egg have already come together to form the cell. And that's what mitosis is. Now, 3 reasons why cells reproduce. Growth, the ability to grow from an infant to an adult. Repair, replacement. Again, all these things are vital and important to the body, and it happens at the cellular level in mitosis. Now, there are the different phases or the different stages of the cycle of mitosis, and again, it's a looping cycle, and once it gets through one cycle, it'll go through a second time and a third, and it continues. The phases are interphase, prophase, Metaphase, Anaphase, Telophase, and cytokinesis. Now, interphase and cytokinesis truly technically are in there, but they're not really considered the main 4. And the main 4 are prophase, Metaphase, Anaphase, and Telophase. Now, in your Edmodo, I have some questions for you to answer as you go through the lecture. And then one of the questions is, when does mitosis begin for an animal? And again, we answered that. It happens after the joining of the sperm and the egg. And again, it's asexual. Interphase is the first phase. And again, it's one of those phases that are not technically in mitosis, but this is where the normal cell is seen. And we have the nucleus, we have the chromatin, and in class we talked about how chromatin is basically unwound DNA that is, when it winds up, it'll become a chromatid, and then when it makes copies of each, it will be called a paired chromosome. And we'll see that as we get going. We also have the membrane, nuclear membrane, and then we have the centrioles or the spindles. Now, in interphase, we go from interphase to understanding what is happening. We see the chromosomes are copied, and they start to copy. They're not totally finished until prophase. And then we look at the DNA is making exact replicas. So each chromatid, each side of a chromosome or a paired chromosome is the exact same as the other. Now here are just 2 cells of interphase, and it's really tough to see interphase and prophase, really the first 2. It's really tough to see them at the cellular level. Now prophase, we see the spindles start showing up. And again, the spindles start going to the opposite ends of the cell itself. The nuclear membrane starts to disappear as we look at that as well. And the chromatids have made copies. Now this is why in many ways that prophase is considered one of the most important, because of that copy feature. If it's not that copy feature, then we would keep getting less and less chromosomes as we reproduce our cells. But because of that copy feature, all our cells are going to be the exact same. There are the chromatids. Now in prophase, again, it's the first step in mitosis. It's where cell begins to divide. The centrioles or the spindles appear, and they begin to move to the opposite ends, and the fibers start to really show up. In prophase of the cellular level, we don't really see it again. It's a little tough to see what we're looking at here, but the next few phases we will definitely see it. Now metaphase, you have this equator. You have this equator right here in the middle, and this is an imaginary line where all the pairs of chromosomes line up on the equator. So again, that key word there is on. Later on in meiosis, you're going to see a little bit of twist to metaphase, and that's what causes different variations and us to have variation. But as we see here, we have 4 original chromosomes on the bottom, and then their copies are on the top. and the spindle fibers are attaching, and they're starting to line up on the equator. Now again, what is happening? The chromatids, or pairs of chromosomes, attach, and then the pairs line up on the equator, and that's in metaphase. Now in metaphase of the cellular level, you can actually see it. You can see the chromosomes pairing together and lining up in the middle. Now it leads us to anaphase. Anaphase, we see the centromere, or the bridge, the middle of the chromosome split. So now in the upper part of the picture, you see 3 chromatids that are the exact same 3 chromatids as the bottom, but they're split apart now. And now they're being pulled to opposite ends of the cell, and it's with such a force that the cell starts to split. So we move from anaphase, and again you see anaphase at a cellular level. You actually can see the cell pulling the chromosomes from one side to the next. And then you get to telophase, which the nuclear membrane starts to show back up. And then the fibers start to go away. And as you look at the edges of the cell, you're going to see that it starts to create this little furrow, this little area where the cell starts to split. Now again, the cells don't necessarily split and fall completely apart here. They will stay together if it's for a body, for any kind of body cell. And then again, forming of a body, it'll stay together. In class, we talked about how identical twins at this stage, on the first time, if it splits completely, that's where we get identical twins. So there's the cell splitting, and then the nuclear membrane returns. So again, our steps here: the nuclear membrane returns, it comes back in telophase, and the chromosomes appear as chromatin. They start to unwind again back to the chromatin that we saw at the very beginning of the entire lecture. In the cellular level, we see 2 separate cells now. We see the difference between the 2. That leads us to cytokinesis. Cytokinesis is where the cell does its job. And again, that's where the white blood cell is to seek and destroy the viruses, or the stuff that enters into the bloodstream. Again, cell membrane moves inward to create 2 daughter cells, each with its own nucleus and with identical chromosomes. So now we have 2 cells that have created that are exactly the same. Again, in your Edmodo question quiz, what stage is the most important to cell division of mitosis? Well, in many ways, it's the interphase and prophase. It's that prophase stage that they're all copied by, that allows this whole process to even exist, and life, and really in many ways life and body cells to exist as well. Again, this is just a review: interphase to prophase, prophase to metaphase, lining up on the equator, metaphase to anaphase as the pull apart, and then you have telophase as they're apart and the nuclear membrane starts to come back. And this is where we see it go back into interphase, to where the 1 goes to 2, the 2 to 4, the 4 to 8, and continues on, especially in a human, where the 1 cell goes to millions inside the womb. Again, this is just a plant cell. Plant cells, the only thing is different is that the cell will split and form that wall back in. So that way the cell wall stays intact. Again, just another visual of how the cell cycle is a continuous cycle, and how it moves from one to the next, and showing, if we start in the upper right behind the yellow, the chromatids are there that makes the copies in the prophase. Then we have metaphase that line up, anaphase, they pull them apart, telophase, the nuclear membrane comes back, cytokinesis, they're doing their job, and then it moves back into interphase and does all over again. Again, the last thing is, what is the cell theory rule that this goes with? And that answer is very simple. It's the third that we talked about, where new cells are produced by existing cells. Now, this is the lecture on mitosis. Friday upcoming, we will have the lecture on meiosis.